So once again, hello everyone, and welcome to our MissionLakes webinar. My name is Tallent Dadi. I'm working at the Helmut Center for Environmental Research in Germany, and I'm also the coordinator of one of the projects that is organizing this webinar series. Put my phone on silent. So once again, I want to welcome you all to this MissionLakes webinar that is organized by 4 projects: EUROLakes, FutureLakes, ProCleanLakes and FERRO. These are projects that we funded under the Horizon Europe in 2023. And we are working together on the protection and restoration of Europe's natural lakes. And the term Mission Lakes, I want to say a little bit about it. Maybe probably you've heard about Mission Ocean. So the idea of the Mission Lakes was, this term was actually developed or coined by Lawrence Cavallo, the coordinator of FutureLakes. The need for this was to highlight the importance of lakes and to make a stronger visibility of lakes within the Mission Ocean framework. So we, as the 4 projects, we are working together to share knowledge, innovation, and practical experience. So we've been doing this webinar series since beginning in August 2025, which we had a webinar focusing on innovation in lake restoration. And in November, we had a second one, which was hosted by the ProCleanLakes on entrepreneurial interest in lake protection. So today we have the 3rd series, turning pollution into resources. And the next one will be in spring, which will be hosted by EUROLakes. So I'm very happy to have you all, and I also want to extend my special welcome to our 5 experts who are going to give the presentations today on the topic of turning pollution into resources. To kick off, maybe I just need to highlight a few issues, organizational issues. We will have 15 minutes presentations, and if there is time at the end of each presentation, we will have short questions pertaining to that presentation. And then we move on to the next presentation. And then at the end, we will have 20 minutes for discussion and maybe questions pertaining to all the presentations. So you have 2 options to ask your questions. You can use the chat, just type in the chat, or you can, at the end of each presentation, raise your hand through the tools for raising the hands. So with that, I would like to first introduce our first speaker today, who is from representing the EUROLakes project. He's Professor Raffaele Pellerosso from the University of Tuscia. Professor Tuscia, the floor is yours. You can, Professor, sorry, Professor Pellerosso.
Thank you.
You can go ahead. Yeah.
Thank you very much. I'm going to share my screen. Please let me know if you see my screen. Thank you.
Your screen is good in presentation mode. Thank you.
Perfect. I'm very happy to be here, and I want ready to present part of the EUROLakes project, in particular focusing on the demo site of Vico Lake. A brief resume the project, just to be sure that you know the topic, and we are a group of team. In particular, the coordinator is wetlands in Netherlands, and we are University of Tuscia, that is the work package leader of the demo site, work package number 3, located in Lazio region in Italy. You can see here the 3 lakes that are part of the demo sites: Lake Dumer in Germany, Lake Bistret in Romania, and Lake Vico in Italy. We know I skipped this part just because we have no time, and I want to briefly introduce the concept of EUROLakes, that is considering the entire landscape, engaging all the stakeholders, creating a resilient ecosystem for the benefit of people, biodiversity, and climate. We adopt for a methodological approach that is the four return frameworks that evaluate and consider the returns of social, natural, financial, and inspirational benefit. The community of practice, so the construction of a community, local community by the participatory approach and the integration of modeling. So we have advanced model system impact models inside digital twin approach and the ecosystem service evaluation. Looking at this is the main framework, and the demo site Lake Vico is a particular volcanic lake located in central Italy with a max depth of more or less 40 meters, around 50 meters. The altitude is 510 meters, and the size 12 kilometers square. You can see that the Lazio region has a lot of volcanic lake. In particular, here we have the Vico Lake, but maybe you know also Bolsena Lake, one of the big volcanic lake in Europe, and the other lake, smaller. But Vico Lake is a particular system that we can consider more or less close, because the basin is in the top of the mountain. And we can see here, for example, that there are a lot of agricultural area and forest area, but we can better investigate after later in the next slides. The main issue is the eutrophication that was driven and is driven by total phosphorus and the erosion process. We know that we should have a concentration of 20 millimeter for liter or microgram per liter of phosphorus, but we are over these thresholds, and we have some cyanobacteria blooms, and also we have a concentration of arsenic that is but is a natural concentration for the area. So we aims to reduce the total phosphorus as the main driver of the eutrophication process. And in particular, we have if we have a look up the trend in this parameter in the last years, we can see that there is an increase, in particular in the bottom of the lake, and this is can be a result. Can result in internal loading, a release of phosphorus due to absence of oxygen in the bottom. And there is also an increase of vulnerability and concurrent risk of cyanobacterial blooms and accelerated eutrophication. And so EUROLakes project is really important, assume a strategic role, because we need to reduce absolutely external input in these systems. We adopt the nature-based solution approach, but with the integration of four return returns that consider all the stakeholders in particular. So in this slide, you can see the evolution of the landscape, the land use change from the 1954 to nowadays, and the main increase is in hazelnut orchards that is now the one of the main culture in the area, not only inside the basin but in the Viterbo province. Is a particularly vocated environment, and the agriculture is now the main driver of economy in the area. So we have to balance this aspect, agriculture and conservation. You can see here the main change in the area, and in particular the increase of hazelnut archer that now is around 1,000 hectares in the area. We have also a small area, urban areas in the south part, but also chestnut grows in the higher slope. So pressure, maybe you know already the typical pressure that we have in a lake, but to have a short resume, we can say that land use, erosion process are the main driver of the eutrophisation, but also climate change is important because of the stratification of the lake, the absence of oxygen in bottom layers now is very important. We have to consider it. We have also some wastewater inputs, but not now because the municipality, one of municipality now act to remove the wastewater inputs in the lake, and the main issue still remain the agriculture. In the same area, we have different also the presence of military area, but now is no more a problem, and the increased frequency of extreme rainfall now is also a problem because increased erosion and the readability of the soil. From the impacts are a level of ecosystems, a level of landscape, reduction of habitat quality, landscape aesthetic value, the drinking water supply is reduced, and we cannot drink the water of the lake more, and the municipalities in the area just this year and the last year adopt a different way to capture water from the ground with a lot of investment because of the increase of cyanobacterial blooms and cyanotoxin production. But the eutrophication and the landscape degradation is a cause of environmental degradation, but the other issue, the other theme that I want to underline is that without a good communication and a governance, sense of community, every action can be not effective in the reduction of eutrophisation runoff. We know the issues and the solution from since the 1990s. And the main issue now recognized is the social capital, if we can say, the capacity of the stakeholder to cooperate. And in this project, we work a lot to reduce the conflict between agriculture and tourism and visitors, between land use and conservation, put together people and identify a common governance that can be accepted from both farmers, visitors, tourists, and restaurants, and so on. So we proceeded with a stakeholder mapping of all the stakeholders in the area. You can see now a short image, an image of the global stakeholder mapping that we did. And inside the project, we also have to realize some activities in Vico Lake to reduce, to increase the management, the effectiveness of the management by nature-based solution. Here is the experimental side inside the basin. You can see the localization here. We have a retention basin where all the flow, the runoff arrive, and we have an automatic sample to capture, to gather sample, water sample, video cameras, and the possibility to analyze the water sample in this area to really understand the contribution of agricultural basin in the pollution of the lake. Some action, the monitoring by video cameras, and through the video cameras we can monitor, for example, in the same area, runoff events, linking them with the critical rainfalls, critical events. Here, some images from one of the last event. This is the automatic sample in the area. And also the some weather station that we now have 3 weather station in a strategic point into the basin. And here are the our laboratory and equipment we bought with EUROLakes. And also we have to realize some nature-based solution, buffer areas, permanent grass in hazelnut plantation, drainage channels, retention basin. But to do this one, we have before speak with people through workshops. And in the last, I'm going to present now. And in the last section, number 7, is to integrate the action outcomes in management plans in the area. So, 2 workshops, we realized 2 workshops in this year, the last years, in 2025. And the first one was focused only on farmers that are the main actors of the area, to create trust in us, in the process, in EUROLakes, in university, to start a collaboration that was really necessary to have a successful action in the area, because all the land is private land. If you want to realize something, we have to involve the landowners. And the second one is another, so sorry for in Italian, but we adopt the Italian language, local language, to have a maximum impact on, and so we are going to shaping ideas, share the alternatives. We identify some alternatives and propose them to the people and discuss with them. And to increase the participation and also to gather all the opinion of the people, we create with Fondazione Eni and Enrico Mattei, our partner involved in participatory planning. They develop a tool, a platform, where the people online during the January, and this January, have opportunity in a week to evaluate their opinion and put their opinion regarding the action, the alternative we selected with the support of local expert. And this action are the current management that, because we also have to underline that many things have been done by farmers, but the people that usually are going in the lake don't see these efforts. And there is a conflict because they think that farmers are not going the right way, but the same and the other hand, the farmers don't want people inside the land because obviously different objectives. But so we are going to show what they did, the best management practices they apply, and evaluate them as a current management. After that, we propose the optimization of the grass cover in hazelnut orchards to reduce runoff, environmental conservation and restoration action like rebuilding dry stone and terraces, vegetative filter strips and so on, and nature walk and eco strip along the coastal, removing or rewilding the crops in the shoreline, along the shoreline. And the people have the opportunity to see the results directly of the votation with the support of this participatory tool, decision support systems. And now we are going to, in the next month, we have the results. I don't have the results now about this one, but we are going to realize the most shared alternative in the area to demonstrate the feasibility under the point of view of reduction of impact, also cost and production of ecosystem service. But regarding the topic of this webinar, turning pollution into resource, in the EUROLake, I talk about what can be a pollution and what can be our resources. Lake sediments maybe can transform in a resource for agriculture, but so we have to think about this. This is not part of our project. Other projects are more deep in this. We have a lot of pruning residues that should be now is a pollution because they increase the runoff through block of the natural drainage systems, and they are burned. Can be another source of energy. Several studies in the area. A stakeholder also told me that they want to invest in this type of transformation from pollution into resource. The coastal farmland that can be transformed and changed as a habitat or eco traits. But the problem is not only the material environment, but also how the system is managed. And looking at the social capital, I can see that the social degradation is a personal opinion, is an endogenous pressure of the lake degradation. And if we have degraded social capital, we have also a reduced collective action adaptive capacity. We need to increase this type of social capital, but not to increase because it is already present. But we have to say in the last slide, reactivate the social capital to restore lake ecosystems, because making the social capital visible, we increase the networks, reduce conflict, we reduce the knowledge gaps, and in short, we can say that we have a return of inspiration of the people that now are not so involved in the management of the lake. There is a lack of sense of community and tourists and visitors in another. And without activating this social capital, maybe, and every action we apply is going to have no success. So this is in the slide you can see the contact. I'm the work packager 3 leader, but the scientific coordinator is Nicoletta Ripa, and also we have our collaborator, two PhD students, Chiara and Giulia, that works in this project, and I'm very happy to networking with you and share solution and have your feedback for the next days, the next years of project that are now going in depth, realizing nature-based solution action in the demo sites. Thank you very much.
Okay, thank you very much, Professor Peloroso. Since we are running short of time, I'll just allow one question which is in the chat. The question is from Simon Tanok. The question is, are there any efforts being made to change the dominant algal profile from cyanobacteria to diatom dominant growth to improve internal nutrient processing?
No. So effort there. So many studies, many people opinion about this, but the problem is that I don't have here the slide about the profile, the layers of the presence of oxygen. We have now new data about the content of oxygen in the lake, and in the last 20 meters after the summer there is no oxygen, and so half of lake is without oxygen, and is really a problem. We have to think about it, but I'm here also to have new project to have the possibility to identify solution that can be applied in the. Sorry, I cannot. Yes, I want to see you in my screen.
Are you done?
Yeah, sorry, I'm
Okay.
lost in the screen.
Okay. Okay, so thank you very much, Professor Peloroso, for the interesting presentation on Lake Fico. If there are still any questions,
Okay,
please put them.
yeah, sorry.
Put them in the chat. We will address them at the end of all the presentations. So now I want to invite a group of two people. So that is Dr. Olga Tamauk from the University of Helsinki. She's an associate professor, and Dr. Kasper Reitzel from the University of Southern Denmark. He's also an associate professor. So they will give two closely related presentations. The floor is yours, Olga.
Hello to everyone. My name is Olga. I'm a researcher from University of Helsinki, and today, together with Kasper, we have a great pleasure to represent the FERRO project at this meeting, and we will share our experience with sediment nutrient recycling. And as you can see from the title of the FERRO project, this is quite of central importance in this collaborative initiative. So why is recycling of nutrients, and especially sediment nutrients, is so important? It is largely because of we are concerned about eutrophication, and eutrophication is not only an issue of European lakes. It's a global problem that is reflected in proliferation of blooms of cyanobacteria, in loss of biodiversity, in high greenhouse gas emissions, and high economic costs. Eutrophication of lakes has accelerated with changing or altering natural phosphorus cycle when we started mining phosphorus for food production. By this, we have enlarged phosphorus cycle, and this phosphorus cycle, human altered phosphorus cycle, is much bigger and it is terribly leaking. Managing eutrophication is getting increasingly challenging, but because there are no drastic changes in nutrient flow to foresee in the near future, there are relatively limited opportunities to reduce further point source pollution, but also reduction of diffuse pollution is very challenging. Climate change works against, but importantly, we have huge accumulations of phosphorus in watersheds. And nowadays, it is almost impossible to improve ecological conditions of lake without addressing internal phosphorus load. Here is one example of hypertrophic lake in southern Finland, Lake Enäjärvi, for which we showed that internal phosphorus load drives long-term changes in eutrophication. But we provided evidence also for that internal phosphorus load explains also spatial variability in eutrophication across multiple lakes in southern Finland. And here is even more evidence from around the globe in this book, and we have just heard in the previous presentation another example. But eutrophication is not the sole issue we are concerned about. We are also worried about the situation with phosphorus rock, which is a non-renewable resource, and this is highly unevenly distributed across the globe, with most of deposits concentrated just in a few countries of the world. And Europe almost entirely depends or relies on the import of phosphorus supply. So Europe is especially vulnerable. And that is why it's time to think about closing phosphorus cycle, and it can be done especially efficiently by sediment removal from lakes and by recycling sediment nutrients as fertilizers. So this is because by sediment removal, we will also reduce this important internal source of pollution. And here I would like to highlight another Finnish case, the case of Lake Galtrask, where sediment removal at very restricted area, in fact, reduced considerably phosphorus concentration in the lake, but also promoted decrease in cyanobacterial blooms in long term. And I truly believe that there can be even more success stories like this. But there are quite a few studies showing also ineffectiveness of sediment removal in lake restoration in the conditions of high external nutrient load. And this is not surprise. In fact, it's just these cases just remind us about the importance of system analysis. So it's important to, first of all, to ascertain which drivers, which stressors cause eutrophication and what is their contribution. Additionally, sediment removal has always been argued for high costs and for high ecological disturbance. But here I would like to highlight the potential of targeted sediment removal, or a potential of focusing restoration sediment removal efforts just on limited area, on hotspot areas. So this could be a sort of cost-efficient solution with little disturbance to environment. And there are also novel technological developments, of course, to highlight that can potentially reduce environmental disturbance of sediment removal. But finally, we shouldn't forget this great potential of repurposing sediment nutrients as fertilizers. And my experience with sediment nutrient recycling started with the studies of Lake Mustijärv. This is a small lake located in Estonia, and this is the first well-documented case when the effort of sediment removal was combined with sediment nutrient recycling to agriculture. So the lake was fully desilted from sediment to increase, at the first place, lake depth. But already in 2 years after sediment removal, we found that pool of releasable phosphorus was rebuilt. And this resulted also in pronounced internal phosphorus load. And in fact, this created a unique opportunity to learn from the next sediment removal experience as a part of the FERRO project, but this will be already another story at one of the next webinars. So the effects of sediments removed from Lake Mustijärv on plant growth were studied in mesocosm experiment with ryegrass and in the four-year field experiment with the mixture of grasses. And under controlled conditions in the mesocosm experiment, we observed that sediment addition improved both plant biomass and phosphorus uptake by plants. And generally similar observations or similar results were observed also in this four-year field experiment. So this was our first and very positive experiment with experience with sediment nutrient recycling. But if we check literature, we can find also opposite effects. And in fact, literature review is not really helpful in providing explanation for such a discrepancy and in clarifying factors that determine sediment phosphorus availability from sediments. And most of the studies so far are just pod studies. These are predominantly pod studies with sediment of one lake. But major problem is that sediment in these trials is applied as growing media, so not as fertilizer, not according to the content of the phosphorus in sediment, which potentially increases both to environmental risk of both nutrient leaching, but also of contamination with heavy metals and other pollutants. And for this reason, we carried out another study, another experiment, testing sediments from multiple lakes, from 4 Finnish lakes and 2 Estonian sediments. And in this experiment with growing ryegrass on sand, we had treatments with no phosphorus addition, with phosphorus added with mineral phosphorus fertilizer, and with phosphorus added with the different lake sediments to meet application rate of 25 kilos phosphorus per hectare. Additionally, we tested amendments like lime, biochar, and herbospolar mycorrhiza for their potential effect on improve bioavailability of sediment phosphorus for crops. So, according to our results, in all cases, sediment increased plant biomass, and we see that sediment supplied plants with phosphorus, but also with other essential macro and micronutrients. Additionally, we found that both lime and biochar amendment improved plant growth, increased plant biomass, and especially in the Kymmijärvi sediment treatment. So there can be also interaction between sediment and amendment. And most of the sediments that we tested in this experiment, they had elevated content of zinc. Most of the Finnish sediments, and this was in this sense they differed from Estonian sediments. But notably, lime, biochar, and especially lime reduced zinc content in plant biomass and reduced it substantially, so that enabling the use of the plants as feed for domestic animal species. So this is very important finding. And we continue to explore best practices of sediment nutrient recycling now as a part of the FERRO project. And in our new experiment established just a couple of months ago, we test 6 sediments of 6 lakes in southern Finland. Sediment recently removed from Lake Mustijärv, but also phosphorus flocks produced generated at the restoration process at Röde Reservoir in Germany. And we use all these materials as phosphorus fertilizers. In the experiment, we test also again biochar and lime and the phosphorus solubilizing bacteria to potentially improve phosphorus availability for crops, but also to reduce environmental risk. And study design is quite similar to what we had previously, but this time we are more focusing on mechanistic understanding that will brings us closer to this best practice of sediment nutrient recycling. And for that, we are doing measurements. We continue to do measurements and sampling to characterize plant growing conditions until May 2026. So stay tuned. But also a few concluding remarks for my part. So my experience shows that both sediment recycling and both sediment removal and sediment recycling as fertilizers has a great potential in eutrophication management, but this potential is really underestimated at the moment. Developing best practices and more success stories will certainly improve the situation, and we are working hard in FERRO for that. So, and one important concluding remark also here is that the potential of sediment nutrient recycling should be in fact considered or taken into account already at the quite early stages, at the stage of system analysis, so that it will be possible to design the solution suitable for each particular case. Thank you very much, and now my FERRO brother Kasper will continue the story.
Yeah, but first of all, thank you, Olga, for introducing the whole concept of why we should do this lake restoration by sediment removal. So that makes it a bit easier for me to go straight to some of our experiences with the lake restoration by dredging. So since the last 5-6 years, we have been part of a big national Danish project with the many research institutions involved, with the focus of first of all restoring this Lake Ormstrup, a small 3.5 meter deep lake in Denmark. As we can see, the lake is surrounded by fields, so it has of course received nutrients from the catchment, but there has also been extensive stocking with ducks during the 80s, which really have added or accelerated the eutrophication of this lake. So when we look at the sediment depth profile, we don't need to go into details, but we can see total phosphorus, which is the sum of all these colored bars here with depth, is especially increased in the upper layer, the upper 20 centimeter of the sediment. So the purpose of this big project has been to first of all understand the lake. So we have spent many years and efforts, multiple partners, looking into sediment biogeochemistry, water chemistry. There has been digital twins developed, fish monitoring, etc. So we have a lot of background knowledge, and the idea has then to be and develop a gentle dredging system, so we could remove only the uppermost sediment layers, which in this case would, just shut this down, mean that we would remove around 6 tons of phosphorus, which we would like to recycle in the agricultural catchment. So that is the main purpose. So here's the results. That is what we call the lake butt dredger. That's a pump-driven dredger. So this has been designed during this project. And if we see here, before it goes into the water, here we have the suction head. So the whole thing is pumping up sediment. And the idea is that this suction head is adjustable, so we can adjust it to always cover the uppermost layer of the sediment. And there's a little video from the early days underwater where you maybe, it's not the best, but can see a little bit of how it works. Down here you have the suction head. So the whole focus is that we want to make it a very gentle way with a low amount of disturbance in the water. So the muddy sediment gets sucked in here, pumped through this yellow hose on land. Here we have a storage tank, but we also have a system where we add polymers And if you look here to the right, the reason why we add the polymers is to flocculate the sediment, so we ensure a faster and more efficient dewatering. So here you can see after, in this case, a synthetic polymer has been added, we create larger sediment particles, and this enables a faster dewatering. So the sediment and the polymer goes into these dewatering bags or geobags. You can also see one down here. And here the water is slowly draining out of these bags within a few months' time. This water is collected, and unfortunately we didn't have a picture with the sand filter, but we also have an iron-coated sand filter in order to trap particles, but also excess phosphate that could be in this reject water before it goes back into the lake. So the result is that we end up with a nice portion of mud, this very nutrient-rich but also organic-rich sediment after it has been dewatered. And this is what we have done quite a lot of studies on during the last years, especially my former PhD, Sina Hasler, she's here, has worked a lot on different types of tests with the whole purpose of evaluating the phosphorus fertilizer value of the sediment. So it has ranged from laboratory scale to small scale field trials to larger scale field trials, which I'll just talk about now. So just to remind you, this is the sediment phosphorus profile. We do these sequential extractions. We don't need to go into details, but the important finding is we look at this light orange part. This is phosphorus bound to iron, and this actually makes up a large part of the surface sediment that we get up. More than 50% of the phosphorus is bound to iron. So therefore, one of the first experiments we did was a laboratory experiment where we wanted to test the fertilizer, especially the phosphorus fertilizer, value of using sediment as a substitute to a commercial fertilizer. So TSP here is the commercial triple superphosphate fertilizer and inorganic fertilizer that farmers use. So I didn't show it here. We also have a zero control with no phosphorus added, but then we added sediment. We added sediment where we actually pre-extracted it and tried to remove the iron-bound phosphorus. And we also made our own iron phosphate, and all of them were dosed. So the important thing here is that they are all dosed, so they have a similar total phosphorus concentration. So we can compare what the sources of phosphorus might differ. And then we established a P fertilizer replacement value that is basically to evaluate, first of all, how much biomass did we produce by adding the triple superphosphate compared to a zero control. And then we compared the efficiency of adding sediment or the different iron compounds. And what we see is that we get not as high a fertilizer replacement value as the triple superphosphate, but roughly 60% of the increase in biomass was observed when we had the sediment, much less when we use sediment where we have removed the iron phosphate, but slightly more actually when we used our synthetic iron phosphate. So this indicates that the IMP is actually seemed to be relatively available for, especially for the barley in this experiment. So we also wanted to look into what happens if we have no plants in our soils. So we tried to assess the phosphorus mobility in the soils, and here's the setup. Basically, we have some small columns, and they are made up in a way so we can make an inner substrate you see here. And this is where we have our P source. This could be soil fertilized with triple superphosphate or with sediment. Then it's separated from surrounding soil by a membrane, and we then moist it up and leave it for roughly a month. And when that is done, we can slice our soil into very fine 1 millimeter intervals. And what we then do is to just do a simple water extraction of this soil layers to assess how much phosphate has moved from this inner substrate into our soil. So if we look at, first of all, this is first, if you see here, the x-axis indicate the distance from the substrate. So here is the 1 millimeter closest to the substrate, further out to 12 millimeter away. So if we add no phosphorus and simply just have soil in the middle packed with the same soil around it, we can see there's no really tendency for dissolution of or solubilization of phosphate from the substrate. Then we add triple superphosphate fertilizers in different ratios. 30 kilo is what is recommended for the farmers in Denmark. That is the maximum. And what we see is when we increase the concentration of phosphate in this substrate, we can see we start to dissolve and see a migration of this phosphate into the soil surrounding the inner compartment. And this increases even more the higher the dose we have. Then if we try to compare to our sediment, here we compare the 30 kilo per hectare. And then it's quite interesting to see when we use sediment as a phosphorus fertilizer, we see absolutely no signs of solubilization of phosphate. So in this case, it simply stays in this inner compartment. This is the same for the sediment where we try to remove the iron phosphate, and more or less the same when we make our own iron phosphate fertilizer. There's a small tendency here. But a general finding is that we can say it seems that the sediment is relatively available for the plants, but when we have it in the soil, it's much less soluble than the triple superphosphate fertilizer, which is a nice feature because that could reduce the leaching, for instance. Then we also try to upscale this a little bit to these 1 square meter field studies. Again, using the sediment from Lake Ormstrup, we tried to do 2 different treatments where we flocculated the sediment or dewatered it by either a biopolymer or a synthetic polymer. And we also made a kind of a biochar out of it, the HTC, and we compared it, of course, to the triple superphosphate again during 2 growing seasons. And now we use the ryegrass instead of spring barley. So here, just to show to the left, the light blue here is a control with no phosphate added. And we have our sediment dewatered by a biopolymer, by a synthetic polymer, and the biochar based on the sediment. And here we have the commercial inorganic fertilizer, the triple superphosphate. So it's very obvious that at any sampling dates, when we measured water extractable phosphorus, as how much phosphate is in the pore water more or less, we could see the tendency is the same as we showed before. We see a higher dissolution when we use a commercial inorganic fertilizer than when we use sediment as a fertilizer. And also to just show you that it was not because the phosphorus was not available. Here we see dry biomass compared to the control. We can see all the treatments here with sediment or with triple superphosphate showed more or less the same increase, and also for the phosphate uptake. So again, we can support the conclusion that it seems that the sediment is available for the plants, but it does not leach into the soil as easily as a triple superphosphate fertilizer. An even larger scale, now it was 24 square meter of plots. Again, we have done many different treatments, but here we just focus on comparing the 30 kilo per hectare phosphorus fertilizer by a triple superphosphate and compared it to our sediment. So here you can just see sediment has been amended here in the setup. And then instead of having ryegrass or spring barley, we used pea as a crop here. And this is how it looked when it was more or less ready for harvesting. Then we collected everything. And if we look at the total biomass, we could see, compared to the control, we saw a tendency for increased biomass when we used both the sediment as a fertilizer and the superphosphate. Antonio, his postdoc working on this, also did some parallel studies on these plants with the sediment again. Here we have also used biostimulants. These are the dark colored symbols. For now, we just ignore them. That's another story. But if we look at the open symbols, that shows here we have the control, we have the sediment, and we have the triple superphosphate here. And what this result showed was, again, that total biomass increased when we used sediment as much as when we used the commercial fertilizer. We could see especially the shoot biomass was where we saw the major increase. And interestingly, we could see a lower, or at least a tendency for a lower root biomass when we use the sediment compared to similar concentration of the triple superphosphate. If we look at other effects than just the phosphate and the biomass, then we can see signs of added benefits, actually, where we can see reduced leaching potential, and that is based on here where we see the Olsen P. That is what can be extracted in the bicarbonate buffer is actually lower here. We could see the biomass was similar, but we again see a tendency for a lower leaching or solubility of the phosphate that are in or is in the sediment. We also evaluated the cation exchange capacity, that is the ability to retain other positively charged nutrients mainly, and we could see that is also higher when we apply sediment, and we also see a higher water holding capacity. So these are also positive aspects in terms of soil fertility, and that is most likely because we also apply a lot of organic matter. We have humic acids and might also have clays that have these negative charges and retain cations, but also give a good structure of the soil. So just to conclude here, we are in the coming years following up on this. We have established new field plots that we will follow for 3 years periods. Again, different doses of sediment and compared to different doses of commercial phosphates. And we also work more on these biostimulants to see if we can see efficiency, more mobilization of phosphorus when we have biostimulants, and look into different dosing strategies as well. So this is something we will hopefully get more results on in the coming years. Then a question is also, is it risky or can it be safe to reuse sediment in agriculture? Because as Olga also mentioned, there might be other elements than just nutrients in this sediment. So one of the things we have been looking into here is a survey from 10 Danish lakes where we collected surface sediment. We measured heavy metals and in general found not the zinc to be an issue, but instead cadmium was, at least here in 6 of the lake, above the limit that you're allowed to bring on the field. This does not mean that you cannot bring it, but it means that you have to bring it either over many years or more years, or you have to bring it on a larger area, so you remain under this limit. So what we are looking into now in the coming years is if there are any bio-based solutions where we could try to reduce the potential risk of having, for instance, too many heavy metals or PFAS or other harmful substances in our sediment. One of the things we're looking into is to use industrial hemp for phytoremediation. Hemp is known for like a really rapid growth, taking up a lot of water and mobilizing a lot of elements in the soils. You can see slides down or papers here indicating that it has been efficient in mobilizing heavy metals, but also PFAS, etc. So our focus will now be, can we, for instance, use hemp to remove heavy metals and PFAS in our geobags before we bring it on the fields. So we will be testing whether we can maybe have a growth season or 2, and in that way extract the nutrients into these hemp plants and have a more safe disposal of our sediments. Just the final slides, more or less, is our future plans. We have received some funding to continue this dredging of Danish lakes, and also with a focus on developing on this lake bots. What we especially are going to do is to try to see how cheap can we make sediment dredging compared to comparable restoration tools like chemical precipitation. So our hope is that we can develop it so it can be comparable in costs to, for instance, a chemical treatment. We will also compare if we can combine it with a small amount of P binders, so we also take out the nutrients or the phosphorus in the water column. As I mentioned before, combine the geobags, the dewatering, with the phytoremediation to extract potential harmful substances, and then of course look into the recycling on the fields. So hopefully in the coming 5 years there will be a lot more activities here in some of the Danish lakes. So in conclusion, I think we can say that definitely the sediment shows nice potential as a fertilizer and also as a soil improver. And I think it is especially interesting that when we have a lot of iron-bound phosphorus, we see that we have lower tendency for leaching and solubilization of the phosphorus in our soil. And then, of course, we should be aware about potentially harmful substances, and hopefully we can show that nature-based solution might be able to ensure a safe reuse. So thank you very much, and thank you all to the rest of the group. We are many people working on this, so yeah, thank you.
Okay, thank you very much, Olga and Casper, for the interesting presentation. We are running short of time, but I'll allow to have the questions that were asked in the chat. The first one was to Olga. Actually, it's two questions. The first one is why 50 or 100 centimeter of surface sediment removal? Is it loosely binded? And then how the new surface sediment conditions are changing? That's the first question. B, what about benthic vegetation biodiversity with dredging?
Yes, thanks for the question. That's a good question, and I think that it was partially answered by Casper also. But in general, I think the question is about like Galtrask that I mentioned. In that case, in fact, the sediment was removed to increase lake depth. So there was not prior analysis of sediment before that. So we do not know it was not. The intention was not to, there was no intention to clarify the forms of phosphorus, but basically, yes, the idea is to remove this potentially mobile phosphorus that can be potentially mobilized and participate in the eutrophication. But yes, so we do not really know which form of phosphorus we removed, but I just can't say because the case was successful. I can just think that there was a lot of this potentially mobile phosphorus or maybe iron-bound phosphorus. And further sediment stabilization would likely need like additional measures, perhaps some let's say food web interactions, perhaps, and like milder methods that could promote further improvement in the ecosystem. And what else? And there was
a question. There was a question about, I think, some biodiversity and how you
biodiversity, and this is exactly the point that Casper raised, that there are opportunities of smart dredging or gentle dredger application, but there are also, as I mentioned, what we are doing, we now are focusing on the potential of this targeted sediment removal. So not to produce this disturbance at the whole lake ecosystem scale, but focus on just limited area so that disturbance could be minimum.
Maybe I could add, I would also say that for the lakes we are looking into in Denmark, it is actually pure mud. So I'm not, to be honest, very worried about disturbing life or plants or anything in the sediment, because there is no macrophytes. It is really eutrophic lakes that we're looking into at the moment. So I would still say I would believe that an improvement in the water quality will definitely improve biodiversity in the long term compared to small impact you might do by removing a lot of mud that consumes oxygen and releases greenhouse gases and all this. So I don't see that as a big challenge, actually.
Yeah, and I would like to add also, because my experience also shows that, in fact, the sediment removal at the Smustijärv, it was totally degraded lake, but afterwards we observed decrease in biodiversity. So this is like a gradual improvement, but it just needs more time to be visible.
Okay, thank you. The other question was probably, Casper, you can start, was from Raffaele Pelleroso. What potential options exist for the recycling or beneficial reuse of sediments in a lake deeper than 40 meters, such as Lake Vico, without disrupting ecological balances? I also question whether the associated costs would be economically sustainable.
Yep. I think that is a nice difference between the Italian and Danish lakes, because in Denmark we have so many smaller ponds with only a few meters of water depth, and we only have a very few lakes that has 40 meter. And as the system is right now, we would never start dredging a 40 meter deep because the equipment cannot handle this. And in that case, I think, like Olga says, there are more potential in looking into deeper areas that might be a major source of pollution in such a lake, and see if there was some ways of dredging. But for now, this dredger system would not be at all economically viable in such a deep lake. So it is, at the moment, we are more focusing actually on getting it able to dredge more shallow lakes than now, because the limit is 2 meter. So we want to get into more shallow water, because I think we have more than 150,000 small ponds in Denmark, where you could really improve biodiversity by improving their condition. But I agree, it's not a good solution as it is now to dredge a very deep lake. We cannot do that.
And I would like to add, we also in Finland, most of our lakes are shallow, so dredging is like more an option here. But I think that what can be working better in your case is hypolimnetic withdrawal, perhaps. And Casper is also working on the recycling of phosphorus removed from hypolimnion as a fertilizer. So this can be an option in your case.
Okay, there's just one last question. I hope you can answer it quickly from Lawrence. I have a question whether you are applying plowing in order to mix sediment with soil in field application.
We did not do that in our case. It was just applied after soil preparation. And then we manually mixed it down a little bit, but not like plowing it deep into. We more or less followed, I think it was the same, we talked to the farmers where they, for instance, would apply manure on the fields.
We are using mixing we mix, in fact, we also in our lab studies and in the trials in the lab studies that should mimic conditions as much as possible as it will look like in the field conditions. And for example, we mix sediment to about 5 centimeter layer. That would be like a more practical way of distributing sediment or mixing sediment into soil.
Thank you very much, Olga and Casper, for the exciting presentation. Now we have to move on to the contribution by FutureLakes, who is represented by Dr. Margarida Costa from the Norwegian Institute for Water and Research. Margarida, the floor is yours.
So thank you. I think you're seeing my screen now. So good afternoon, everybody. I'm gonna move a little bit out of the lake topic, but I'm still gonna keep on the waste resource utilization topic. I'm talking on behalf of FutureLakes, which is a project that is coordinated by Lawrence here at NIVA. But also this one I'm going to show locality. It's another Mission Ocean project that we coordinate as well. So just I'm going to give you a brief context of the project. How does it appear? And I'm going to go through the project and some of the results we have, focusing on the ecosystem we have established here in Norway, the industrial ecosystem. So the project comes within the Mission Ocean, which has a major goal. I think everybody in the room is aware of the big goals of the Mission Ocean, of restoring ecosystems and biodiversity. It's within the lighthouse in the Baltic and North Seas, and has two main phases, one of deployment and piloting, and another one of development and piloting, and another one on deployment and upscaling, which is exactly the one we are entering right now. So in this context, the main goal of locality, the project locality, is to develop circular and sustainable value chains linking key industrial players to stimulate co-creation and bring new and innovative algae-based products to the market. So our main goal is to bring products out to the market, and within this goal, we aim to establish microalgae cultivation systems linking the different industries, promote industrial cooperation, promote circular economy, ecosystem services, and so on until we reach the products, develop those products. But we don't want to stop there. We don't want to stop at the development phase. We want to have the products out in the market, and therefore consumer acceptance and prepare the deployment of those products. So this is how the concept of the project looks like. The project is using seaweed harvested in the Baltic and North Seas, but also microalgae that it's cultivated using waste streams from 3 different industries. So the textile, the aquaculture, and the greenhouse plant production industries have nutrient-rich waste streams that are suitable for algae cultivation. So what we did here in the project was to aggregate those industries in a way that the waste suppliers can supply that waste, that for the microalgae cultivation industry, it's a resource. So those biomasses, both the wild seaweed biomass and the microalgae cultivated using the streams, they are either used in bulk or biorefined into different ingredients for product formulation. And here at the end of the scheme, you can see that we develop products that will feed directly the same markets that supply the waste stream. And that's what we call the industrial ecosystems. So we try to give back to the same market sectors products, the same market sectors that supply the waste streams to develop or to grow that microalgae. So there's a huge difference going a little bit into the algae cultivation. There's a huge difference between cultivation up in the north and in the south of Europe. So the fundamentals are pretty different. In the south of Europe, where the industry is quite well developed, the industry is focused on outdoor cultivation, larger areas, and a lot of sunlight that it's available. So you can see here on the right side that the farms are big and outdoor. However, in the north of Europe, where you don't have so much sun naturally available, the farms tend to be smaller. Especially for that reason, they have to be indoor and use artificial lights, and for that reason they can also occupy smaller areas. And for that reason, as it is a high resource intensive type of cultivation, it's key to establish this industrial synergy. So this is key not only for waste utilization, for treating this wastewater that would otherwise needed to be treated in the plants, but it's also key for the economic feasibility of the algae farms, the microalgae farms. So in the project, we have these 3 ecosystems that are located in countries where the markets are key for the economy of those countries. So obviously in Norway, we have the aquaculture ecosystem. I'm going to show you some results that we have obtained within this ecosystem. But we have the aquaculture, the agriculture / food ecosystem, where there's a lot of wastewater coming out of greenhouse plant production that is established in the Netherlands and Belgium. And we also have the textile ecosystem operated in Sweden. So this is only possible because of a huge consortium that it's mainly composed of industries, since the industries are the key stakeholders into this ecosystem establishment. But this is also based in a lot of research and development. So there's a lot of research groups involved. And as you saw from the beginning, so this project goes from algae cultivation. So there's experts covering all over the value chain. And in the different sectors, so from algae cultivation to product development in the food sector, textiles, agriculture, and all the sectors that I have mentioned. So just very, very briefly, I think you can see the video here. So our initial approach was to collect side streams from all the industries I mentioned. Those are the ones that NIVA, these tasks are the ones where NIVA is focusing, where we have a biggest role. We collected the streams from all of those industries. Here you can see this is a small production farm. You can see our partners, Shasha, which is a researcher also contributing to FutureLakes, collecting the wastewater. Wastewater was collected in different points of the farm so that we could analyze. And this was done for the aquaculture sector, sorry, something happened here, but for the aquaculture sector, for different farms, it was also, sorry, this was unexpected. It was also done for the fish processing industry. So we are also collaborating with an industry that is processing fish residues that allows us to utilize some of their waste streams as well. So just this is not to take a deep look into the numbers, but it's just to give you an overview of how complex those streams are, because they are all very rich in nutrients that algae can utilize, and that's our main focus here. But then they are very diverse, and one main challenge that we find is that the composition is not standardized. So even though we collect the waste stream in the same season, in the same place, in the same farm, there's always going to be variability, and that's the main challenge we deal with. So you can see an overview of all the chemical analysis that we have done to the different streams here, and you can see this is just for you to have an overview of how complex and how diverse those streams can be. So what we do here in EVA is try to combine those waste streams with an algae that can grow, an algae strain that can grow in that particular stream. So an algae that has nutrient requirements that are somehow similar to the nutrients available in those waste streams. So here in EVA, we own a big culture collection of algae. We have in-house more than 2,000 strains, which makes us the perfect partner to develop this kind of assays. So we bring the streams in, and then we select between the 2,000 algal strains we have in-house. We select the perfect one to cultivate into those streams. So we run these assays in these 96 well plates that allow us to produce big amounts of data in very short time. And just so you have an example here, this is an example we run for one of the waste streams. The main partner involved was very much interested in food applications. So he wants to establish a food production line And therefore we screen all the novel food-approved strains that we have in the collection. You can see all the chlorellas; those are all chlorella strains. All chlorella strains that we have in house, and we screen them using those 96 well plates and managed to identify the two best performing strains. So the ones that presented a higher growth rates, and therefore we could explore further the potential of cultivating them in those waste streams. So we took those strains to a higher scale. We also have a pilot cultivation facility in Iva, where we can cultivate up to 1,000 liters of microalgae. You can see here a 5-liter cultivation, where we managed to, so the blue line is for the standard culture media, and the red one is for the waste. And you can see that the waste is allowing for higher growth rates of this particular strain that we identified, that we were able to pick from the collection. So the growth rates, the growth rates are pretty similar. The end, the final biomass was higher in using the waste stream. We also, because this is for food application, so one thing that is quite important is that the final biomass it's suitable for this application, and the chlorella is mainly used for food, and because of the high protein content. And you can see that the biochemical composition does not change if we use standard medium or a waste stream. So this is also, I think you can see, this is also a video. I don't have any results yet to show you because this was run last week. But the partner has a few bioreactors, and we went on proof of scale or proof of ecosystem to their facility, and we received wastewater directly into the bioreactors that he has available, and we took the algal strain from Niva here, and we managed to run the reactor for a week and collect some biomass out of this reactor. So we managed to prove that this can happen also at the scale on site. Okay, so just to remember a little bit about the project. So we were covering the first part of the project. I'm just gonna browse very quickly so that you can have an overview of what it's being developed by partners in the project, and what is the biomass being used for. So without going deep into the results. So many producers are also involved. Algae producers, seaweed and biomass are also involved in the project, with the goal of uptaking the innovations that are being developed, but also to supply the biomass that it's needed for the product development, while we don't have enough biomass for that. So you can see there is several partners around Europe, and also biorefinery pipelines are being developed for several biomasses or groups of algae. So this one here that you see, it's a pipeline that was established for diatoms, which we found that had a fraction with very interesting bioactivity into our aquafeed trials as well. And then we have the work packages dealing with product formulation, and very quickly you can see here that the agriculture work package is developing biostimulant and biocontrol products. They have just produced the prototypes to be used in the field trials this summer. So those were the prototypes. Just they are ready now for the main trials. They have shown interesting activities, are seaweed based, and have shown interesting activities in lettuce and tomato, promoting the growth under stress, under drought stress. And biocontrol as well, and against pests, several pests. Also, you can see on the right part that textile that was colored with spirulina. So the textile partners are mainly interested on dyes, natural dyes, and antimicrobial additives to be applied into, for instance, sport clothes, so that the clothes don't get as smelly. But we also have partners working on aquafeeds. I just mentioned that Diatoms has shown interesting results, protecting from sea lice infestation, protecting the salmon for sea lice infestation, and also several food products, mainly protein analogs. You can see an omelette done with white chlorella, for instance, some fish analogs, and also in the right side you can see sausage-like type of formulation that it's going to be prototyped also this summer. So just to finish up, because I mentioned in the beginning that the project also has a huge market and social component as well. We work a lot into consumer acceptance, because we want all the products that we are developing to reach the market next year. So not all the products, but some of the products should be out in the market. So we work a lot on integrating the acceptance and the consumer perspectives into the development of those products, but also adjusting the formulations according to market expectations. This you can see the video on our website. Sorry. And also we work a lot in the business part. So checking consumer barriers, legal barriers that go throughout the different market sectors that I mentioned, not only on algae cultivation, but throughout all the sectors. Economic barriers. So we have partners that are supporting the SMEs involved on assuring that they have the what it's needed to deploy the innovations. Also LCA analysis and business plans. So just to conclude very briefly, this is of course only possible because of all the partners. The consortium is a very big consortium. We are 27 partners. This is only possible because these industries have taken this initiative of coming together and teaming up in different areas that are very different for all of them, but where these knowledge gaps can be key on bringing new innovations, new products, and new processes to the market. So to finalize, this is the consortium. I want to thank the entire consortium involved in the project, a very big group of people, but also my team here at NIVA, these people on top of the picture that have been intensively working on the results I showed you today. So thank you so much. You can look for locality social media website and find a lot of information there. Thank you.
Okay, thank you very much, Margarida. I don't see. Is there a question? Okay. Okay, it's a comment. So I don't see any questions. But maybe I'll just ask one myself. I see that there are a lot of industrial partners involved in the project, which is interesting. My question would be do they already have some products that they are on the market, or
Yes.
is it only the products that are developed in the project?
No, some partners have some products in the market. So it's not only SMEs, it's some well-established industries, big industries involved as well. Some other products I'm trying to improve, some other partners I'm trying to improve products that they have already in the market and try to integrate with the solutions that are being developed here in locality.
Okay, thank you very much, Margarita. So we have to move on to our last presentation, which is being presented from the ProCleanLakes by Professor Tina Economy Amili from the University of National and Capodistrian University of Athens. Professor Tina, the floor is yours.
I'd like first to thank Margarita for her lecture, which is a very good introduction to my lecture, which is more academic. The focus of this lecture is the ecological and economic value of unique microalgae found in oligotrophic lakes. Algologists are continually searching for new taxa, often expecting to find them in extreme biotopes, whether defined by harsh physical conditions, seasonal occurrence, or simply because they remain understudied. Freshwater biotopes with high potential for taxonomic discovery include ephemeral waters that often exhibit extreme conditions, benthic habitats, especially biofilm-associated communities, groundwater systems, and environments characterized by unusual temperature regimes, such as thermal springs or tropical regions, as well as other atypical habitats. It's widely recognized that nutrient-poor oligotrophic waters, such as clear mountain lakes or granite-based silt lakes, play an important role by providing low competition environments that support the persistence of slow-growing and specialized algal taxa. Let us now examine in more detail the function of microalgae in oligotrophic lakes. First, microalgae serve as highly sensitive environmental indicators due to their narrow ecological tolerances. Shifts in species composition can signal environmental changes such as climate fluctuations, land use alterations, or atmospheric nutrient deposition. In addition, changes in abundance or morphology may reflect recent disturbances, including warming, altered mixing regimes, or subtle nutrient enrichment. And do not overlook that sediment records of certain microalgae, such as diatoms, preserve historical signals that allow the reconstruction of environmental conditions over time scales ranging from decades to millennia. Taken together, these characteristics make microalgae valuable biological archives that connect present-day observations with long-term ecological history. Moreover, microalgae play a critical functional role in maintaining ecosystem stability in nutrient-poor water systems by efficiently recycling nutrients, supporting higher trophic levels such as zooplankton and fish, and regulating water quality through oxygen production and carbon sequestration. Moreover, they are indicators of ecosystem resilience. Resilience refers to ecosystem's ability to withstand disturbances without degrading. Thus, presence and diversity of microalgae reflect not only current ecological health, but also the lake's resilience to future stressors. Microalgae enhance ecosystem stability through functional redundancy, rapid responses to environmental change driven by short generation times, and their role as early warning indicators of impending ecosystem degradation. Not to forget the conservation value of microalgae in protected lakes. The microalgae often include rare endemic or relict species that have survived after stable, low-impact conditions. These communities preserve irreplaceable genetic and functional diversity, maintain natural reference conditions essential for detecting anthropogenic change elsewhere, and safeguard biological indicators critical for long-term monitoring and adaptive management. Moreover, by examining microalgae, researchers can integrate insights into past environmental change with present ecosystem processes and future resilience. This perspective underpins evidence-based conservation efforts and reinforces the importance of safeguarding oligotrophic lakes against nutrient enrichment, climate stress, and biological alteration. In the last years, microalgae, recognized for their capacity to sustainably produce value-added metabolites, are drawing heightened scientific and industrial interest and should be formally integrated into alternative development strategies. They exhibit high metabolic diversity, producing a wide range of compounds, including polysaccharides, omega-3 fatty acids, proteins, vitamins, and biopigments. They grow rapidly, achieve high productivity with efficient photosynthesis, and can be sustainably cultivated on non-arable land using wastewater and CO2 while requiring few resources and allowing year-round production. Their capacity to produce a wide range of value-added metabolites positions them as important biological resources for future applications in food, nutraceutical, pharmaceutical, cosmetic, bioenergy, and other industrial sectors. This potential is further enhanced through advances in biotechnology and biorefinery approaches. The microalgal diversity does support local economies. Through ecotourism, income can be generated while raising awareness of relevant ecosystems, and the revenues can be reinvested into habitat protection through monitoring projects. Moreover, microalgae provide valuable services such as water purification, nutrient cycling, carbon sequestration, etc. Payments for ecosystem services or public-private partnerships can provide compensation to local communities for their stewardship roles while avoiding the over-exploitation of natural populations. Microalgae-focused educational programs, research stations, and citizen science initiatives can attract funding, students, and partnerships, while training local residents as technicians, educators, or research assistants builds human capital and embeds conservation principles within economic development. Moreover, microalgae biodiversity represents a valuable source of novel compounds for applications in food, cosmetics, etc. To safeguard ecosystems, access should be regulated through permitting systems, environmental impact assessments, and benefit-sharing agreements aligned with biodiversity and genetic resource legislation. Cultivation should prioritize control systems such as bioreactors or closed ponds over wild harvesting, thereby minimizing pressure on nature habitats while generating skilled local employment. This type of consideration as an alternative development strategy was also presented at Greece Lake Trichonis, one of the 3 demonstration lakes in the ProCleanLakes project, where microalgae have likewise been neglected as promising avenue for science-based sustainable development. This lake is a protected one by the international legislation, including the Natura 2000 network and the Ramsar conventions and other environmental agreements, And its management unit operates under the Greek Ministry of Climate Change and Environment. Trichonis is a warm monomictic lake, the largest lake in Greece, belongs to an extended lake district made of 5 natural lakes influenced by Acheloos River, the flow of which is regulated by 3 dams for the production of hydroelectric power, irrigation and food protection of downstream areas, and preventing overflows to the lakes. Geotectonically, this lake is a depression, and the surrounding rocks are the semi-permeable fleece, which is less dense to the north, where apart from the fleece, limestone formations with very high water permeability also found, and very sparse to the west of the lake, where fluvial formations are found. This geotectonic background helps explain why Lake Trichonis remains its oligotrophic status, despite pressures from tourism and agricultural activities, such as olive oil cultivation and exploitation. Lake Trichonis holds substantial socio-economic value, with its primary use in agriculture and secondary use in recreation and tourism. External threats to water quality are linked to olive oil mill wastewater, despite being officially classified as controlled, while internal pressures stem from sediment processes within the lake. Fish and gastropods are among the endemic or critically endangered species. The area also supports interesting taxa of birds and higher plants, with a special emphasis on species from the wet meadows. Endemic phytoplankton means microalgae, particularly diatoms, were also reported there. Various initiatives have taken place in Trichonis aimed at its environmental conservation, and targeted programs have been implemented related to the community engagement, spatial planning, water management, even related to cultural and recreational events. It is worth noticing a successful recent intervention. Recently, a major company planned to establish a pumped storage energy unit in Lake Trichonis. In response, local residents and various organizations acted swiftly, forming a cooperation of 33 associations and agencies in the region, which submitted a legal notice to the European Commission. And the European Commission formally conveyed its negative position to all authorities that had issued a favorable opinion on the project, including the Ministry of Environment, which had declared that it could not provide an assessment. Recent campaigns within the ProCleanLakes project framework helped pinpoint the most effective water quality sampling stations in Lake Trichonis and showed clear seasonal trends in phytoplankton concentration, peaking in water and early spring, and declining in summer and autumn. Water quality indices, combined with historical and recent data, suggest that nature-based designs for Trichonis should prioritize nitrogen reduction and reduce pesticide use. In contrast, geoenvironmental assessment indicated that the unique carbonate-rich sediments and the relatively stable water chemistry constitute a distinct system that requires customized conservation strategies. The suggested nature-based solution approach with local microalgae is focused on cultivating them in lab photobioreactors before transferring them to ponds for bioremediation, while also offering potential for innovative biotechnological uses based on their properties. The process so far encompasses all steps from algae isolation and culture to large-scale development in photobioreactors. Furthermore, all required steps of biomass extraction from the isolated species, followed by analytical chemistry procedures, will be conducted to characterize the chemical compounds for potential biotechnological applications. To summarize, protected oligotrophic lakes support highly specialized microalgal communities that thrive in nutrient-poor conditions and contribute significantly to ecosystem stability, water quality, and biodiversity. Although biologists often study these organisms for their ecological importance, they are frequently underappreciated, as with lake trichonies, as a potential resource for sustainable science-driven development based on microalgae. Therefore, dialogue between scientists and decision makers should focus on co-producing knowledge, translating scientific findings into decision-relevant evidence, integrating science into policy frameworks and management tools, establishing structured platforms for exchange, emphasizing long-term socio-economic perspectives, and building capacity and preserving institutional memory. Considering the scientific evidence, ecological importance, and potential for innovative nature-based solutions using microalgae, the designation of Lake Trichonies as a demonstration lake within the ProCleanLakes project, to my opinion, is fully warranted.
Thank you, Athena, for the interesting presentations. Yeah, thank you very much. So the idea was we are only left with 10 minutes. So I quickly ask in the round, are there any questions to all of the presenters, that maybe some questions that have come up after the 4 presentations that you want addressed? Simon is asking a question. In the world of turning pollution into resources, how much work is being done to improve water quality by driving eutrophication back into food webs via improving or changing internal cycling, and thereby creating improved fishing, improved tourism, and water quality? Such process creates greater production of products, i.e. edible fish, tourism visitor days, and better quality drinking water, not necessarily creating resources, but certainly creating positive financial outcomes. So, yeah, who wants to take that one from our experts? Olga, Raffaele, Caspar, Margarita, Athena.
Okay, maybe I can take this question, but I don't know how much. We have discovered this or discussed a lot about this in publications about sustainable lake restorations. You may be aware of those publications. So it's called blue economy, or creating. We discussed like potential of potential benefits, core benefits of lake restoration by sediment removal. So the idea is that by improving lake water quality, we also improve, we get some other potential core benefits, and we stated like it's something that needs further advancement, and there are some certain knowledge gaps in technologies and things so on. But working certainly, we are certainly in this direction, I would say. And I think today's efforts have nicely demonstrated this, that we are working on this through at least integrating circularity into lake restoration or water quality management.
Thanks, Olga. Just to, I'm not sure if I'm allowed to just jump in and speak without writing a question, but I guess my question relates to obviously, you've talked about the 150,000 lakes within the Danish area there, and obviously the deeper lakes aren't going to be suitable for those sorts of situations. So how many or are there other systems or researchers looking at the reduction of phosphorus and nitrogen in systems, which through nature-based systems, which then create those, by obviously having better water quality, you've got greater fish production, you've got reduced numbers of cyanobacteria blooms, which are going to limit tourism days, and for any of these water bodies that are also bringing in or being used for drinking water or raw water storage, that's obviously going to have an impact on processing costs of activated carbon and things like that. So I was just wondering, is anyone within the group actually actively doing that? Because that's something that I'm working on, but I'm just wondering if anyone in the group is doing that sort of work.
I'm not fully sure I understand, but what I can say is that, for instance, we're also looking into to more sustainable solution addressing like the water by these solar-driven pumps. But we also have worked intensively with the socioeconomics with this emphasis of really trying to figure out what is the true values of doing lake restorations, other than just improving the water quality, because often it's a costly thing, and it would definitely be nice to define, as you also mentioned, what else, what is the actual value of having a clean lake with respect to greenhouse gas, with respect to like public perception, all this. And a lot of this, I'm not an expert, but I definitely know it's very difficult to assess these values. But I think it's really needed, because if it's only a cost-benefit analysis of how expensive is the dredging, and that's it, then it's often a lose-lose. And also, if you compare it to the cost of phosphorus, it's really not easy to argue that by dredging you save a lot of money on phosphorus fertilizers, for instance. So but I think all of these measures we're doing with restoring lakes is with the hope of improving the water quality and the fishing and all this. But how you really nail it and really put, for instance, a value on it. I know from some Dutch studies very nicely that it was a very easy thing, because they could do a chemical treatment. Water was nice, no blue-green algae. People came and swim, and they were paying a ticket for entrance to the lake to swim. So it was a good case for this lake owner. And every third or fourth year, where the lake was going a bit back, it was still possible to pay for this treatment, because it gave a positive return. I don't know if that answers your question, but
it sort of does, I guess. Like, I've done sort of back of the envelope calculations with our system of treatment. We've seen sludge reduction, we've seen increases in biodiversity, increases in fish size, things like that, and just on reductions in fugitive greenhouse gas emissions, treatment can start to basically, on carbon credits, if you could carbon credit it, pay for itself within 3 years, and then after that, it's just its actual income plus the fishing benefits, the tourism benefits, the water quality that I speak of. I mean, that's the sort of thing that I'm really, hoping to find out more about, and the methodology is simply to steer the microalgal populations from cyanobacteria dominant to diatom dominant, which was my earlier question. And in doing so, you're extending the period of time before nutrients are recycled, because diatoms will move nutrients either into sediments or up through the food web, rather than the cyanobacterial, rapid recycling that occurs, that also happens to benefit themselves. So by reducing the cyanobacteria populations and moving the nutrients through diatoms and through the food web, you're sort of capturing both of those benefits.
Yeah, and in fact, we have many knowledge gaps to do this cost-benefit analysis. For example, we just can assume that, for example, if sediment removal or some other restoration is effective, we can assume that we reduce, for example, greenhouse gases, but evidence is still very limited. We need real data to prove this and to do life cycle assessment. And in terms of sediment recycling, as I mentioned, we have really many knowledge gaps because this information, the topic itself is very fresh. We are producing like absolutely novel knowledge. We are coming with new knowledge. How to do this? What are the effects, environmental effects, and how exactly apply the sediment to the field? So it's something that really influences also this cost-benefit analysis and life cycle assessment, after all.
Okay, so in the interest of time, I would want us to move quickly to the next step. But first, I want to thank all our 5 experts for their insights and for this nice, interesting presentation that they gave today. So thank you very much to all of you. Would be hosted by EUROLakes. It will be in the end of April or beginning of May. They will share the information. So please look forward to the emails we will share through the networks. And this will be another exciting version of this webinar series. Anka, do you have something from ProCleanLakes you want to share?
No, only to say that it was very interesting today, and I thank you to the speaker from all projects. So no, I don't have anything.
Okay, thank you, Anka. Lawrence, do you have something to say? Lawrence, are you still with us?
He had something in chat, so.
So probably he left. Okay. So, I mean, since Lawrence is not there, then I also want to take this opportunity to thank you, all of the attendees who attended today. It was a long webinar, but I think it was a very interesting and exciting. And once again, thanks to the to our experts, our 5 experts. And I wish you a nice afternoon and hope to see you in the future versions of our webinars. We will be going with a rhythm of every 2 to 3 months. We'll be having a new webinar with an interesting theme and interesting presenters coming up to share their knowledge. Thank you so much.